sperm, human sperm. So after the spermatogenesis, so we have studied the production of sperm, which is taking place in seminiferous tubules. So what happens? The production is of three steps: multiplication phase, growth phase, and maturation phase. In multiplication phase, all the spermatogonial cells are produced from the germinal epithelium cells. Okay, those are spermatogonial cells, are the germ cells or sex cells which undergo multiplication, that is mitosis. Within that, they are divided into type A and type B cells. Type A cells, what happens? They can renew themselves and they can multiply the number of spermatogonial cells in one end. Another end, type B cells, they undergo the complete spermatogenesis process. A single spermatogonial cell produces 4 sperm at the end of the spermatogenesis process. We know from the spermatogonial cells, they undergo growth phase, they increase in their size and converted into primary oocyte. Primary oocyte undergo first meiotic division to form 2 secondary spermatocyte, not primary oocyte, sorry, primary spermatocyte, and they get converted into 2 secondary spermatocyte by undergoing meiosis 1. Okay, after this they undergo meiotic second division, meiosis 2, and each secondary spermatocyte divided into 2 spermatids, means totally 4 spermatids. Now what happens? The division stops. This spermatids undergo differentiation or metamorphosis and get converted into sperm, okay, or spermatozoa. And this is the structure of spermatozoa. So what happens when they get converted? Spermatozoa, spermatids are single celled, simple cell which get converted into, yes, specialized structure called as spermatozoa, and that process we call it as spermiogenesis. Okay, so the ploidy of the sperm is haploid, that is the chromosomal set in the sperm is 23 in number. Okay, so the sperm structure basically divided into 3 regions: head, middle piece and tail. Okay, between the head and middle piece there is a region called neck. Okay, what is this head contains? So this head or complete sperm is covered by plasma membrane, a double layered membrane, where in head portion it consists of 2 structure. One is the nucleus, the big large nucleus containing the genetic material. Okay, because we know nucleus contains DNA, that is genetic material. And in the upper portion, tip of the sperm, they contain acrosome. Acrosome is the structure where they consist of some special enzymes, we call it as hyaluronidase. Okay, this is lytic enzymes which helps to digest the wall of ovum during the fertilization. They need to penetrate, right? So how this penetration happens? With the help of acrosome, the enzyme which is present in the acrosome, they digest the wall of the ovum so that they can deposit their nucleus into the ovum for the fertilization process. So this is the head portion. So next portion is neck. At the region of neck, there will be 2 centrioles. One is proximal, the center one is proximal, and these 2 are distal. They are nothing but axial filament which is made up of axial protein. Okay, or actin, sorry, not axial, actin protein, and these filaments are called as axial protein. This is made up of actin and myosin. We know all the tail or flagella portion, okay, where there is a locomotion, those structures are made up of our structural protein, that is actin and myosin. So here also, which leads to the tail portion, right? So which makes the sperm motile. Okay, they are motile. Why they are motile? Because they need to travel from the vagina till the fallopian tube. Remember the structure of female reproductive system. So where is the site of fertilization and where is the insemination takes place? So there is a distance. The distance should be traveled with the help of tail, because how they got such a structural change? With the help of the structural proteins called actin and myosin. Okay, so this centrioles are present in the neck region. Next to the neck region there is middle piece. Especially they are surrounded by spirally coiled. They surround the axial filament. Okay, this spirally coiled structure we call it as mitochondria. Okay, especially why mitochondria? What is the function of mitochondria? Mitochondria is called as powerhouse of the cell, means which supplies energy in the form of what? Energy currency. What is that energy currency called as? ATP, adenosine triphosphate. So here also the presence of mitochondria will give the same reason. Why? Because of when they travel from the vagina till the site of the fertilization, not only they need tail to move, but energy. Okay, that energy will be supplied by the mitochondria, which is present in the middle region, and the tail gets the strength from the axial filament. From the neck region they get fused to form the structure called as axoneme, okay, which gives a strength to the tail region. So this is the structure of human sperm, and it size, like length, will be 50 to 60 microns in length. Okay, we'll study some statistical things. A normal young adult, a male adult, forms 120 to 130 million sperms per day. Per day, how much an young adult means? We know when the spermatogenesis starts, after the puberty only. When a male individual attain maturity, spermatogenesis starts. So during that spermatogenesis, a young healthy adult male, how much of sperms they produce? 120 to 130 million sperms per day. Okay, and the viability means how many days or how many hours they are viable or alive. They live only 24 hours. Okay, so next is during ejaculation, means during insemination process or gamete transfer. We know in humans it is by insemination, transfer of male gametes into the vagina of the female reproductive tract by the process called as ejaculation. Okay, during the ejaculation process, how many numbers of sperms are ejaculated in the female body? 200 to 300 million sperms. Within these 200-300 million sperms, 60% of these sperms will need to have a normal size and shape, and remaining 40% should be vigorously motile, means they need to be active during the motility. They should be very active. So 40% are very vigorously motile sperms, and 60% compulsory should have a proper size and shape. The proper size and shape means sometimes what happens, the abnormality of the sperm I am explaining. Some sperms are without a tail, some sperms are without head. I'll explain you what happens. If some sperms are without a tail, how they are going to move till the site of the fertilization. That is one problem. If they don't have head portion, okay, what happens? Acrosome are the important part which helps the sperm penetrate into the egg. If there is no acrosome, no enzymes are ready to digest the wall of the ovum. The sperm cannot travel into the ovum. So this is the drawbacks, abnormality. So size and shape is very important, okay, for the time of fertilization, during the time of fertilization. And already we know the semen, okay, during the ejaculation they ejaculate semen. What is semen? It is seminal plasma and sperm togetherly, it is called as semen. What is seminal plasma? As we have studied a male reproductive system, along with the accessory ducts there will be accessory glands, that is a pair of seminal vesicle, a pair of bulbourethral gland, a pair of prostate gland. what all these three gland does, they secrete some secretions which can be said to as seminal plasma, which contains especially fructose, calcium, and some certain enzymes which keeps sperms active and motile or maturated. Okay, hence the seminal plasma plays an important role to keep the sperm viable, proper or immature condition. Okay, and what is the pH of a semen? See, the pH is almost neutral to bit basic, lightly basic, that is 7.2 to 7.6. If there is a low sperm count during the ejaculation, if there is a low sperm count, if the sperm count is less than 200 or less than 100, such condition we call it as low sperm count, or it is also called as azoospermia. Okay, medical it is called as azoospermia, which in turn indicates low sperm count, which in turn leads to the male infertility. Okay, male infertility. Understood? So this is what the structure of human sperm. One more important diagram for 5 marks. Okay, practice properly with a neat labeled diagram will fetch you the complete 5 marks. Okay.